Hey Mustangs, in this video we're going to be taking a look at the cell cycle. In order to do this, you're going to need your handout that you got in class, so your key concepts handout that we already filled in the top, and you're going to be filling in the bottom during this video here. So let's go ahead and take a look real quick at a important concept that you really need to know and understand in order to get the rest of what we're going to be looking at here. So we looked at in class eggs. We learned are haploid. Remember, hap equals half, so half of a pair means only one, so they have only one of each homologous pair, never both. If it has homologous pairs, that's not good for an egg. Sperm of the same species have to have the matching chromosomes. So for every chromosome that you see in the egg, there has to be a matching one in the sperm for almost all species out there. Again, for each chromosome in the egg, there has to be a matching one in the sperm, and vice versa as well. Okay, there's a matching chromosome to form homologous pairs. So that when the sperm fertilizes the egg, both of these being haploid, come together. Oh, so there's 4 chromosomes here, 4 chromosomes here. When they come together, they make a diploid cell. When the sperm fertilizes the egg, that very first cell is called the zygote. So we see zygote here, and you can see how all of the pairs came together. So this is a homologous pair. One was from mom. This was found in the egg from mom. This came from the sperm from dad. One in the egg from mom, one in the sperm from dad, and so on for every homologous pair that you would find inside of a diploid cell. And then this cell would divide and create the body cells of the organism. So when we're talking about body cells, another term that we can use for body cells is going to be somatic cells. Okay, so that's another term you're going to hear. So somatic cells are always diploid. When you look at the numbers, 4 chromosomes here, 4 chromosomes there. Here we have 8 chromosomes, 1, 2, 3, 4, 5, 6, 7, 8. So with the cell cycle, we're going to be taking a look basically at the lifetime of a cell. Cell cycle is very basically predictable changes that are going to happen in the lifetime of a cell. And you can even think of a human life when you're thinking about the cell cycle. There's very predictable stages in a human life. We know that there's birth, so for every human there's birth. There's growing up, there's childhood, then there's puberty, which leads into the adolescence, and then finally adulthood, and then eventually death. So just like there's the human life cycle where there's very, very predictable things that you know are going to happen at different points, the cell, each individual cell has a cell cycle. So that's the lifetime of a cell. And during the cell cycle, during the lifetime of a cell, there's very specific events or things that are going to happen during each point of the cell cycle, each point of the cell's life. So first thing we have to ask ourselves is, why would a cell need to divide? There's 2 major reasons why a cell will need to divide. The first is cell division, to cause growth. So cells need to divide in order for an organism to grow. So from birth all the way to adulthood, we add cells onto our body. In order to add cells onto our body, existing cells must grow and divide, and that's how we get more. So when we talk about growth, all growth is adding more cells onto our body. And in order for that to happen, cells need to divide. Second major reason why cells need to divide is going to be to replace dead or damaged cells. So we get injured, and when we get injured, we need to replace those cells that have been destroyed. And then there's natural death of cells, and cells need to be replaced. So as cells naturally die in our body, if cells just kept dying and they never got replaced, we would basically be disintegrating, you know. So all cells that are dead or dying will eventually be replaced in order to keep the organism alive and healthy. All right, so in order to understand the cell cycle, we can use this diagram here. So this basically here is kind of like, again, you compare it to a human life cycle here. This would be kind of like, almost like birth of a cell here, and then all the way around. Not till death, but in this one, it would be winding up with cell division, where a cell divides into 2 new cells, or daughter cells. So you're going to hear that term, daughter cells. Okay. Daughter cells is where you have one cell, sometimes referred to as the parent cell, and it's going to divide into 2 identical cells. these are the daughter cells here. So this is a daughter cell, this is a daughter cell. Not quite identical, I could have done a better job on that. But two identical cells, and those are the daughter cells. So these right here would be considered daughter cells. Okay. So if we take a look at the cell cycle, there's predictable phases or steps in the cell cycle. The first is G1, then you have S phase, G2, mitosis, and cytokinesis. Now the majority of the cell cycle are these three steps here. So these three steps here are called interphase. Okay. So G1, S, and G2, those three phases are part of the main part of the cell cycle where a cell spends most of its life in interphase here. Okay, so interphase is G1, S, and G2. Mitosis then comes next, and then finally cytokinesis. Mitosis is going to be where the chromosomes are going to separate and make sure that each of the two new daughter cells wind up with a copy of each chromosome. And cytokinesis is actually the official splitting of the cell. All right. So during G1, this is where a cell grows bigger. So cells, think about it, if a cell didn't grow bigger before it divided, every single time a cell divided, it would get smaller and smaller and smaller. So here, during G1, cells grow bigger. During S phase, cell continues to grow, but the most important part of this is this right here. So right here. During S phase, this is when you make a copy of each chromosome. This is DNA replication. So if you remember DNA replication, that's where helicase enzymes are going to unzip the two strands. DNA polymerases, ligases, and all that stuff are going to make copies of the two original strands, and you wind up with two identical DNA molecules in the end. That's happening right here. Okay, so this is where you make a copy of every single chromosome. So S phase is super, 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 super important because that's where you make a copy of every single chromosome. Then you have G2. Cell continues to grow a little bit bigger, and this is where it prepares to divide by getting the cytoplasm ready, preparing organelles for division, and all that. Now, there's checkpoints that are found in the cell cycle. These checkpoints are basically spots when the cell is going to stop and check to make sure that everything is happening correctly. If things are not happening correctly, the cell will either stop and wait until something is corrected, or if it can't be corrected, the cell might actually go through the process of apoptosis. So if something's really bad and things can't be fixed, apoptosis might occur where the cell might actually just simply die. So there's three major checkpoints that you need to identify. So it says up here, add these checkpoints to your picture. So there's a G1 checkpoint. This is probably the most important checkpoint. At this point here, if things are messed up, this might cause a cell to stop going through the cell cycle to divide and actually enter into another phase called G0. And that's the resting phase where the cell's not really doing anything. It's not preparing to divide. And during this time, it can actually make repairs. So if there's something wrong here at this checkpoint, it can get sent into G0. Otherwise, if everything's okay, it just continues through. There's a checkpoint here at G2 as well, making sure all conditions are right for the cell to divide. And then even here in mitosis, there's a point where the cell actually checks to make sure all the chromosomes are lined up correctly and connected and are ready to split, so that way that each new cell, each new daughter cell that is created winds up with a copy of the chromosomes, of each chromosome. Now, you have two other boxes on your paper. So once you have the cell cycle filled in, you're going to fill in these two boxes to the right using this information here. So you need to know what it looks like when a chromosome gets copied. So during S phase, so don't forget S phase. Okay, S phase is one of the three phases that is during interphase. During this phase, that's when the DNA gets replicated. So that means you make a copy of a chromosome. So what you're going to draw is, you'll draw a chromosome and show that when you make a copy of the chromosome, it looks like this. So this is one single chromosome. When you make a copy, the copies actually stay connected together at first. Eventually, these copies will get pulled apart. So this here, this is one. Here's the other chromosome right here. And these are identical to each other. So these are identical copies. We call each copy, when it's stuck together, a sister chromatid. So this is a sister chromatid here. This is a sister chromatid here. They're just simply copies of each other. So whenever you have chromosomes stuck together and they are exact copies of each other, we call each copy a sister chromatid. So you'll define this as well, either in your box or on the side of the box. So sister chromatids are exact copies of each other. The point where they are attached together, that's called the centromere. So again, defining the spot where two sister chromatids hold together is the centromere there. Okay, so this is super, super, super important here. Make sure you understand this. So when you make a copy of a chromosome, and I could even do it over here. So when you make a copy of a chromosome, the copies stay stuck together. So this half here would be considered a sister chromatid, and this is a sister chromatid, and they are exact copies. So I'm going to use this image here of a celebrity to help us see that. So here you make an exact copy. And it's almost like identical, not identical, Siamese twins. So it's like Siamese twins. They're stuck together. So this is an exact copy of this chromosome right here. Now, don't confuse this with making a copy of a homologous pair. Okay, homologous pairs and sister chromatids are not the same thing. Homologous pairs are chromosomes where one's from mom, one's from dad. They're the same size, same shape, with genes in the same place, but they're not identical. Sister chromatids were identical. They were exact copies of each other. Homologous pairs are not. They're similar to each other. Okay, so here we have a homologous pair, one from dad, one from mom. I'm not saying that Megan Fox here is a man, but I'm just showing how they're identical. We have two celebrities that look very similar to each other, but they're not exactly the same. When you make a copy of a homologous pair, you make a copy of the one from dad, and the copy stays stuck together. You make a copy of the one from mom, and the copy stays stuck together. So this here and this here, these are sister chromatids. This is sister chromatid, sister chromatid. Both of them are the same exact copies of each other. That's the one from dad. Same thing here, sister chromatid, stuck together at the centromeres. So go ahead, answer this question, and we'll be talking about it in class. How are sister chromatids different from homologous pairs? All right. So last thing, looking at the cell cycle here, remember G1, S, and G2 are all interphase. Basically the cell's growing. Most important step is probably here when it actually makes a copy of all of the chromosomes. And then you have M phase, which includes mitosis and cytokinesis. If you take a look at this cell here, has this cell gone through S phase? So if you look here, has the cell gone through S phase? The answer is no. If this cell had gone through S phase, we would see sister chromatids. Right here, we only see homologous pairs. So this is a diploid cell. We know di means two. It has homologous pairs in it. When it goes through S phase, it's going to look like this. You make a copy of each pair. So a copy, and each pair, remember, one is from mom, one's from dad. One's from mom, one's from dad. One's from mom, one's from dad. One's from mom, one's from dad. You make a copy of each one, and now you have sister chromatids. So sister chromatid, sister chromatid, sister chromatid, sister chromatid, okay, of each one. So whenever you see a chromosome in an X shape, that's the clue that S phase has occurred. So whenever you see chromosomes in the X shape, S phase has occurred. Okay. So here, if we draw a cell, okay, and I put in some chromosomes like this, okay, we have homologous pairs. We know one of these is from mom, one of these is from dad, one of these is from mom, one of these is from dad. If this is going to go through S phase, after S phase, I make a copy of the one from mom. Here's the homologous pair, that's the one from dad, make a copy of that. Now I have sister chromatids stuck together here, sister chromatids there. And then I do the same, if this is the one from mom, make a copy of it. Copies are still stuck together, sister chromatids. Here's the one from dad, make a copy of it, they're still stuck together. Okay, so now we know it's gone through S phase.